Nucleic acid is composed of long polymer of building blocks called nucleotides. Each nucleotide is composed of a sugar molecule, a 5-carbon ribose, and a nitrogen-containing base and phosphate group. In this lecture, we will focus on 4 topics. In part 1, I will introduce you nucleosides and nucleotides. In part 2, I will introduce you structure of DNA. In part 3, we will focus on organization of DNA within the cell. In part 4, we will analyze the structure of RNA. Now let's go to part 1. First, let's look at the base. There are 2 types of nitrogen-containing bases commonly found in nucleotides: purine and pyrimidines. Purine contains 2 rings in their structure. 2 purines commonly found in DNA and RNA are adenine and guanine. Pyrimidines have only 1 ring. Cytosine is present in both DNA and RNA. Thymine is usually found only in DNA. Uracil is found only in RNA. To indicate a specific position in the rings, atoms in the rings are numbered. Basic rules for numbering rings. First, number the rings so that the nitrogens end up with the lowest number combination. As a result, pyrimidine has the nitrogen at 1 and 3. For example, pyrimidine, nitrogen at position 1 and 3. Purine have nitrogens at 1, 3, 7, 9. Here is a purine, nitrogen found in position 1, 3, 7, and 9. Second rule, number the rings so that other functional groups have the lowest possible number. As a result, pyrimidine is numbered so that the carbonyl group are found at 2 and 4. For example, thymine carbonyl group are found at position 2 and 4. Purine, these functional groups are found at 2 and 6. For example, guanine amino group is found at position 2 and carbonyl group is found at position 6. Significant positions: position 5 in pyrimidine. For example, cytosine position 5, related to methylation. Position 6 in purine. For example, the amino group at position 6 will be removed by adenosine deaminase. Defects in this enzyme leads to severe combined immunodeficiency or SCID. There are 2 types of ribose: RNA or ribonucleic acid. Use ribose. DNA use deoxyribose. Here is a deoxyribose. Compare with ribose. Oxygen is not present at position 2 in deoxyribose. RNA is present in the cytoplasm and in high concentration in the nucleolus of the nucleus. DNA is found mainly in the chromosome in the nucleus and in the mitochondria. Addition of a base to a pentose sugar gives rise to a nucleoside. To distinguish the numbering system for the base and the numbering system for the ribose. In the ribose, positions are numbered 1' to 5'. For RNA, there are 4 types of nucleosides: adenosine, guanosine, related to purine, cytidine, and uridine, related to pyrimidines. For DNA, the corresponding 4 types of nucleosides are deoxyadenosine, deoxyguanosine related to purine, deoxycytidine, and deoxythymidine related to purine. Addition of either 1, 2, or 3 phosphate groups to a nucleoside give rise to a nucleotide. They are nucleoside monophosphate, nucleoside diphosphate, and nucleoside triphosphate, respectively. For example, nucleosides derived from guanosine are guanosine monophosphate, or GMP, guanosine diphosphate, or GDP, and guanosine triphosphate, or GTP. The phosphate groups are responsible for the negative charge associated with nucleic acid. Here's a summary of nucleosides and nucleotides for RNA. Here's a summary of deoxynucleosides and deoxynucleotides for DNA. Now, let's move to part 2. The first question we ask is how two nucleotides are joined together. Similar to peptide bond that joins two amino acids, a phosphodiester bond joins two nucleotides. Specifically, a phosphodiester bond joins the 3' hydroxyl group of a deoxyribose of one nucleotide to the 5' hydroxyl group of the deoxyribose of adjacent nucleotides through a phosphate group. These represent acid and these represent alcohol. So the bond between acid and alcohol is an ester bond. Between this acid and alcohol, there's another ester bond. That's why this is called phosphodiester bond. After nucleotides are joined together through the phosphodiester bonds, we have a single-stranded DNA. Here is an example of single-stranded DNA. The end with a free phosphate group is called 5' end. The end with a free 3' hydroxyl group is called 3' end. By default, DNA sequence is written in the conventional 5' to 3' direction. As a result, the sequence of this DNA should be T, A, C, G. Two DNA strands form a double helix. Ribose and phosphate are hydrophilic and they are on the outside. Bases are hydrophobic and they are stacked inside. As you may recall, in collagen, 3 alpha chains form triple helix. A hydrogen bond is important for the triple helix. Very similarly, hydrogen bonds is also important for maintaining double helix in DNA. In particular, hydrogen bonds play a key role in pairing between bases. T pair with A, G pair with C. Under certain conditions, two strands can be separated. The most common form of DNA is B form with right-handed double helix. Between two bases, hydrogen bonds form. Specifically, between A and T, there are 2 hydrogen bonds. Between G and C, there are 3 hydrogen bonds. And this can be seen better in the pictures shown on the right panel. AT and GC. In double-stranded DNA, since A always pairs with T and G always pairs with C, therefore the amount of G should be equal to the amount of C, and the amount of A should be equal to the amount of T. And this is Chargaff's rules. For example, when a DNA sample has 10% of G, how much is the T. Since the amount of C equals G, the amount of C and G should be 20%. The rest are A and T, and it should be 80%. Since the amount of A equals T, T should have the half, which is 40%. In eukaryotic cells, DNA is generally double-stranded, and RNA is generally single-stranded. But there are exceptions. For example, the genome of parvovirus have single-stranded DNA, and the genome of real virus have a double-stranded RNA. This is an example of integration with microbiology. Similar to proteins, in which most alpha helices are right-handed. Most common form of DNA double helix is right-handed B form. Different from proteins, some regions of DNA do exist as left-handed Z form. Two major forces to stabilize the DNA structures are hydrogen bonds, stacking of hydrophobic nitrogen bases through hydrophobic interaction. On the other hand, forces that destabilize the DNA structure is electrostatic repulsion by the phosphate groups. Two strands of double helix can be separated by breaking the hydrogen bonds. In the lab, two common ways to disrupt hydrogen bonds are heating and chemical treatment. For example, in heating, as the temperature rises, the kinetic motion of molecules in solution increases so that the hydrogen bonds are not strong enough to hold. These methods are very useful for Southern blot and PCR. First, let's look at the denaturation of DNA by heating. When two strands are separated, absorbance at 260 nanometer will increase. The temperature at which half DNA is denatured is called melting temperature. Because GC pair has more hydrogen bonds than AT pair, higher GC contents, the higher the melting temperature the DNA will get. There are several ways to break hydrogen bonds using chemical methods. First method is to raise pH. The hydrogen in guanine, for example, can be shredded off when the pH is raised above 10. Second method is to add urea. At high concentration, urea can compete for hydrogen bonding. Third method is to use aldehyde. Aldehyde can modify the amino group and disrupt hydrogen bonds between bases. Now let's switch to part 3. Large DNA molecules must be packaged in such a way that they can fit inside cells. First level of compaction occurs when DNA is wrapped around histone proteins to form nucleosome. Here is the nucleosome. In the center, 8 histone proteins, 2 copies of H2A, H2B, H3, and H4 form a core. DNA wrapped around histone core. Histone proteins contain a lot of basic amino acids, especially lysine and arginine. At the physiological pH, these amino acids are positive charged, neutralize negative charged DNA. Size of nucleosome is about 10 nanometer, linked together by linker DNA. Those linker DNAs are not naked. They are bound by another histone protein, H1. Chromatin at this level resembles beads on a string. Nucleosome further arranged into a right-handed helix or super-superhelix stabilized by H1 proteins. Now, the resulting polynucleosome has about 30 nanometers in size. You may have seen two strings wrapping around each other to make a rope. When you open up two strings at one end like this, you will find the middle part of the rope will become overwinding, leading to supercoil. This process is called supercoiling. Supercoiling happens in bacterial DNA. Supercoiling also happens in human DNA. Polynucleosomes form loops connected by nuclear scaffold protein, represented here in red. Loops form supercoil with a size about 700 nanometers. During mitosis or meiosis, high level of packaging of DNA generates metaphase chromosomes visible under a light microscope. This also happens in the bar body in females with two X chromosomes. Euchromatin is a lightly packed form of chromatin that is enriched in genes, and it is often under active transcription. Heterochromatin is a more highly packed form of DNA, or condensed DNA. Heterochromatin appears as small, darkly staining, irregular particles scattered throughout the nucleus. For example, in this cell, heterochromatin is seen scattered in the middle and in the periphery of the nucleus. Euchromatin is more active in transcription than heterochromatin. Within euchromatin, less packed form is more active. For example, 10 nanometer chromatin is more active than 700 nanometer supercoil. Chromosome abnormalities can be assessed on mitotic chromosome by using karyotyping and by bending techniques. This will allow us to identify aneuploidy, deletion, translocation, and so on. This will be covered in genetics. As you have seen in Chapter 3, genetic information is passed down from DNA to RNA and then to protein. This sequence of events is called central dogma of molecular biology. Transcription is the process in which a DNA strand serves as a template for the synthesis of RNA. Transcription produces messenger RNA or mRNA, transfer RNAs or tRNAs, ribosome RNAs or rRNAs, and non-coding RNAs. Non-coding RNAs are not translated into protein, but they perform specialized structural, catalytic, and regulatory functions. mRNAs make up a small portion of the total RNA in the cell, but they are the key to our production of proteins. In human cells, one mRNA carries information from just one gene. This is called monocystronic. Each mature mRNA contains a coding region, a 5' untranslated region, or 5' UTR, and a 3' UTR. Here is an example of mRNA. Coding regions in the middle, 5' cap and 5' untranslated leader sequence altogether form a 5' UTR. 3' trailing sequence plus poly A tail altogether forms 3' UTR. At the DNA level, the DNA segment corresponding to 5' UTR is located right at the beginning of exon 1, and the DNA segment corresponding to the 3' UTR is located at the end of the last exon. Right here we can see this again in this diagram. This is first exon, and this part is 5' UTR. This is the last exon, and this part is 3' UTR. tRNA are small. They contain a high percentage of unusual bases. They have characteristics, secondary and tertiary structure. For example, there are 4 loops present in tRNA. Each tRNA carries amino acid at the 3' end. tRNA recognize mRNA by anticodon. rRNA make up the largest portion of the total RNA in the cell. They are components of ribosome. They are associated with proteins. Some RNA acts as enzymes, such as 28S RNA. In human cells, there are 4 rRNA species: 28S, 18S, 5.8S, and 5S. These first 3 species, each with a number 8 in it, come from the same gene, which is transcribed into one large precursor and split into 3 species. 5S rRNA come from a total different gene.